Good morning, students. Today we discuss about the topic bacteria. What do you mean by bacteria? Bacteria is a microorganism, isn't it? Yes. So bacteria is one of the microorganism. Bacteria is a cell wall form a rigid structure of uniform thickness around the cell and it's responsible for the characteristics of shape of cells. Here you have to see for some of the microorganisms, that is the classes of microorganisms, that is bacteria, fungi, protozoa, algae, virus and some other organisms. Today we discuss the bacteria. What is bacteria? Bacteria are unicellular organisms belonging to the prokaryotic groups where the organism lack of few organelles and a truer nucleus. Bacteria, why we called as a prokaryotic group? What's the difference between prokaryotic and eukaryotic? Prokaryotic means the organism has a lack of nucleus. It's not well defined nucleus are not found. Eukaryotic organism means the well developed nucleus are present. That is the difference for prokaryotic and eukaryotic organisms. What are the general characters of bacteria? Bacteria is a minute organism seen under the microscope. It's a unicellular organism. It's a prokaryotic. Here there is no chloroplast and mitochondria. It's a lack of cell membranes. Here, what are the parts present in the bacteria? Here, cell wall, capsule, plasma membrane, cytoplasm, ribosome, plasmid, pili, flagella. These are all the parts of bacteria. The cell wall and the cell membrane join together to form a cell envelope. It's important for the protection of the organisms. The cell wall is the rigid wall situated outside of the plasma membrane. It provides the shape of the bacteria and protect the internal organ of the organisms. Then the outer surface, the flagella are present. The flagella are slender and somewhat a minute hair-like structures. They are about 20 nm across and up to 15 to 20 mm long. The eukaryotic flagellum is a long rod-like structures. Already I told the difference between prokaryotic and eukaryotic, isn't it? Prokaryotes means not well developed true nucleus. Eukaryotes means there's a well developed nucleus. So the flagellum is long and rod-like structures in the eukaryote, and the prokaryote is very minute flagellum. It is rigid hair-like structures are present. Here at the end of the flagellum, a trichus are present. So single flagellum is present is called as a monotrichus. Some of the flagellum is found at the one polar at the end of bacteria. Some are present at both the poles of the flagellum. It will be differentiate for the flagellum at one end or some of the bacteria at both end, some of the bacteria at one end, one flagellum. These are the differences for the bacteria. Next, the cell or plasma membrane is a thin structure that surrounds the cell. It is about normally 8 nm in thickness. The structure is a critical barrier separating the inside of the cell from the environment. The cell membrane is also highly selective barrier, enabling the cell to concentrate a specific metabolite and excrete waste materials. Next one is ribosomes. The ribosomes are present inside the bacteria. It is attached to the plasma membrane. Normally, different types of ribosomes are present, 70s, 80s, etc. But in the bacteria, normally found in the 70s ribosomes. The ribosomes are normally made up of protein and ribonucleic acids. Its main function is for protein synthesis. It is support for or transport to the plasma membrane. It is support for the proteins to the plasma membrane outside. Next one is nucleoid. The prokaryotic chromosome is located in an irregularly shaped region called the nucleoid. Normally the nucleoid is made up of the 60% of DNA, 30% of RNA and 10% of protein. The nucleoid is a composed structure. Here the DNA, RNA and protein is present. In prokaryotic contains a single circle double-stranded DNA, but same have linear DNA chromosomes. Next, the mesosome. The mesosome are present in the cell membrane of the bacterial cell that fold inward. It's an important role for cellular respiration. They appear as a pocket formed by the cytoplasmic membrane filled with vesicles, tubules or lamellae. Here, next one is different types of bacteria. First we learn about the general characters. What's the general characters means? It's a prokaryotic organism, microscopic, unicellular, and the lack of true nucleus. Mitochondria and chloroplast are absent. These are the general characters of bacteria. What are the structure of bacteria means? Already I told the pili, flagellum, ribosome, mesosomes, flagella. These are all the structure of bacteria. Next, what are the shapes of the bacteria? The shape of the bacteria is based on the structure, shape and their appearance. Normally 4 types of bacteria. First one is the cocci. Cocci is the round or oval shaped bacteria that is called as a cocci. Next one is rod shaped bacteria we called as a bacillus. Spiral shaped bacteria that is called as a spirella. Next one is a vibrio. Vibrio is a comma shaped bacteria. These are the shape of bacteria based on the size, shape, appearance, nutrition and other structures. Next, how do the bacteria reproduce? Here, can you see that? The outer cell wall, next cell membrane. The cell wall and cell membrane that we called as a cell envelope. Inside the membrane, the elongated nucleoid is present. It's a parent cell. The parent cells or nucleoid cell divides cell wall and membrane begin to form a transverse septum. Can you see the picture in the center portion? The sum of the septum. It's a form to begin the transverse septum. The transverse septum becomes completed. The 2 daughter cells will be developed. Normally, the bacteria has a binary. The reproduction of the bacteria is a binary fission. Bi means 2. When the parent cells are divided into 2, that is called as a binary fission. Most bacteria reproduce by binary fission. In this process, the bacterium, which is a single cell, divides into 2 identical daughter cells. Binary fission begins when the DNA of the bacterium divides into 2. Replicates means it will be multiplication. The bacterial cells then elongates and splits into 2 daughter cells, each with identical DNA to the parent cells. Each daughter cell is a clone of the parent cells. When the conditions are favorable, such a right temperature and nutrients are available, some of the bacteria like Escherichia coli can divide every 20 minutes. This means that in just 7 hours, one bacterium can generate 2,90,152 bacteria. After one more hour, the number of bacteria will be have risen to the colossal or increased it is the rapid multiplication of the bacteria. That's why we can quickly become ill when pathogenic microbes invade our bodies. So can you understand how it will be multiply and causes for beneficial and harmful to our body? It's a beneficial means it will be help of our body. It's harmful mean it create the diseases. I think you can understand the structure, general character, structure and reproduction of the bacteria. Thank you, students.